afternoon, everyone. My name is Shekinah Dosunmu, and today I'm here to talk to you about the effects of male exposure on the neuronal response of the auditory neuron within female crickets of the Acheta domesticus species. So, to start us off, I would actually like to play a small video. Can we get it pulled up? Well, it does not seem like that video is working. But what you would have heard would be cricket chirps, which are the central focus of my research project. So to start off, I'll give a brief outline of my research presentation today. I'll start off by giving a brief overview, some of the background and key terminology and key aspects of cricket anatomy that have already been mentioned. I will then go over into the research projects that have been able to establish the neuronal connection to behavior within the Acheta domesticus species. And then I'll dive into some of the key aspects of my own personal research project. So, for some background, as it has been mentioned already previously, phonotaxis is the movement of female cricket within the species in response to male cricket call. You can have positive phonotaxis, which is movement towards that call, or negative phonotaxis, which is movement away from the call. You can also have no phonotaxis, which is really just no particular movement towards the sound at all. Another important term is syllable period, and where this comes into play is in regards to the male cricket call in question. So previous studies have been able to establish that one of the more important parameters in whether or not the cricket responds to the call is that phenomenon known as syllable period. And what syllable period is it's just referring to the time interval between the consecutive sound pulses of a cricket call. As you can see here in this diagram up here, a cricket chirp is composed of 3 sound pulses, as it has been mentioned before. And a syllable period is just referring to the time interval between the beginning of one sound pulse and the beginning of another. This is measured in milliseconds, and is how we vary the model calls of crickets. Another important term that's key to my research project is that of decrement. So decrement is really a phenomenon that we really only see in one particular auditory interneuron, and that is the L3 auditory interneuron. And what it is, it's just the gradual decrease, not the gradual decrease, but it's just the decrease in the number of neuronal response to each of the consecutive sound pulses. So what that means is that for the first sound pulse, you could potentially have 10 neuronal responses for that first one. But for the very last one, you would have a lower amount, which in this case would be 7. We use these 2 numbers and we plug it into this equation over here, and we're able to calculate percent decrement. The significance of percent decrement I'll talk about later in my research presentation today, but that's basically the key aspect of what percent decrement is. So another key aspects of cricket anatomy that also come into play is that the ovipositor, which is a long extension indicated by this arrow over here that's only present within female crickets of the Acheta domesticus species, and also in other species as well. As you can see, you also have an anatomy of the nervous system of a cricket with the prothoracic ganglion, which houses most of the auditory interneurons that underlie phonotactic behavior located there. The ears are located in the upper thoracic legs, and then the connectors, which ascend to the brain, and which is where the L3 auditory interneuron, the neuron that I'm studying in this project, is located as well. So, the neuronal connection to behavior. So a lot of what I'm currently doing in lab is heavily based on previous projects. So I'll kind of give a quick overview of what those previous projects are. The first is the L3 auditory interneuron. So one of the questions that may be asked about this is, why do we even focus on the L3 auditory interneuron? So some previous studies have been able to establish that when this neuron is impaired in some way or damaged, you see problems with the phonotactic behavior of the crickets, female crickets, in response to model calls in general. So because of those impaired, because of those activities that have impaired the neuron's activity, we know that it's somehow connected to phonotactic behavior within the cricket. The L3 auditory neuron, apart from being able to be connected to the phonotactic behavior of the cricket, has also been shown to respond in a characteristic way. One of which I've mentioned already previously, which is percent decrement. And the other is that it responds to model calls in a particular way of certain frequencies. The frequency that we use in lab is 5 kilohertz, so it also has a distinct response there. The L3 neuron has also been connected to phonotactic behavior within both the Acheta domesticus and a closely related species known as Shirlean Seawood. So previous studies, including the one conducted by Samuel et al., have really been able to establish a starting point for us to work with the female crickets in general. So as you can see here in this diagram over here, this is kind of a representation of some of the data that Samuel et al. Was able to obtain that really connects phonotactic behavior with percent decrement in the closely related species of G. bimaculatus. So on the x-axis, you have phonotaxis and null phonotaxis, with phonotaxis really referring to positive phonotaxis and no phonotaxis being no particular behavior or movement towards the call in general. On the y-axis, you have percent decrement. As you can see between these two graphs, there's a statistically significant difference between the percent decrement levels for phonotactic behavior and that of no phonotactic behavior. And this kind of is the same pattern that we're also seeing in current research that has been conducted within the species we're working on, Acheta domesticus. Other studies have been able to show that environmental variables also play a role in this neuronal connection to behavior and in affecting the neuronal response, with temperature being one of them and neuromodulators such as juvenile hormone 3 being another. Okay, so all of these previous studies that have currently been done or that have been done in the past have really focused mostly on virgin female crickets. This means that the crickets have been from birth isolated from male crickets. They've not been exposed to them. So since we've looked at other research and we've seen that the environment plays a role and can influence this neuronal connection to behavior, one of the aspects of the environment that I'm particularly testing is whether or not exposure of female crickets to male crickets during their growing stage will affect the neuronal response. What that means is that we basically raise these crickets, female crickets, in the same vicinity as male cricket calls. We are right now in the exploratory stages of the project. We are in the process of establishing like fences between male and female crickets so that they're exposed to just pheromones alone. But as of right now, the data that we have is just without that control. So methodology, just a quick review, as it has already been previously mentioned. The key aspect of the research project I'm working on is that necessarily we're recording. So that what that means is that we're recording the neuron's response from outside instead of inside the structures that house the neuron. So first we prepare the neuron. I prepare the neuron by placing it carefully on this wax stand over here so that its two ears positioned on its upper thoracic legs are facing the two speakers, indicated by these arrows. And then after placing it in such a way, I then dissect the cricket to reveal its prothoracic ganglion and its ascending connectives. This is more of a live picture of what we were able to see under the microscope. I then use a suction microelectrode, place it directly on the connectives, and the suction microelectrode is filled with a saline solution that helps us to be able to pick up the neuronal response of the L3 neuron. Okay, so this is some of the results that we obtain directly from just picking up on the L3. So as you can see here, as previously mentioned before, a syllable period is composed of 3 sound pulses. A chirp is composed of 3 sound pulses, and a syllable period is a time interval between those sound pulses. And as you can see on the bottom, you have spikes. So we use this information to generate post-stimulus histograms, which are really just frequency histograms that allow us to be able to pick up on the number of action potentials for each of the spikes that we have. Once we have that data, we then plug it into this equation and we're able to calculate percent decrement. So on a closer look at this post-stimulus time histogram, you'll see that the y-axis is really referring to the bins, so it's what we use to calculate the number of action potentials. And the x-axis is a time interval. So we're able to then use that data and determine how many action potentials are within each time interval and go from there. So from this data that we've obtained so far, I've been able to generate one graph. The bottom graph shows the data from exposed females, while the top graph is a comparison with virgin female crickets and the neuronal response. So as you can see, once again, the x-axis is referring to syllable periods. Here you have 30, 50, 70, and 90. The y-axis is percent decrement. And from the top graph, I'd like to draw your attention to a couple factors. The first is that percent decrement levels range from 30 to 35 for these first three, and then drops quite a bit to 20 for the very last one. The next thing that I'd like to draw your attention to is that of the exposed female crickets. So you're not really seeing such high decrement levels for the exposed female crickets. In fact, their data kind of ranges quite lower, around the 10 to 20, less than 20 area. So you're seeing lower percent decrement at these syllable periods for exposed female crickets. Another key point to point out is that you're also seeing kind of a downward trend within this data set. So there's a slight increase in decrement, but then it keeps on going down. Here you're kind of seeing something similar to that, but you're seeing more of a slight increase in the syllable periods that we would expect to be favorable for virgin female crickets, which is about between 50 to 70 milliseconds. So here there's a slight increase. So in conclusion of the data, we're able to see that there doesn't really appear to be any strong difference between percent decrement across the board for exposed female crickets. It seems that the percent decrement levels, although you do see a slight downward trend, seems to be the same for exposed female crickets. I'll refer back to the graph. But for virgin female crickets, you see quite a difference between at least the 90 and the 1570. So you're seeing a different type of behavior. So from now, we're able to tell that there's a difference between the alpha neuronal response between those two sets of data. However, since no statistical tests have been conducted, we can't say if that difference is significant as of yet. Further studies will hopefully increase the sample size from n equals 4 so that we can more closely match the data that we have from previous study conducted by Ashley, which had a sample size of n equals 10. So I would like to acknowledge Mazel for giving me this opportunity to speak. Andrews University Department of Biology and the Office of Research, along with my research mentor, Dr. Navia. Are there any questions?
So we heard earlier from some of the talks on the phonotaxis tests exposed to males.
Yeah.
And if I remember right, they had an overall lower response.
Yeah.
And that correlates with this, is that correct?
Exactly, yeah.
Okay.
This might be a little beyond the focus of your work, but as a question in terms of actual living male crickets, am I correct in presuming that that mid-range that seems to have the strongest response for the females is where they spend most of their time chirping?
I don't exactly know the answer to that question. I do know that our computer-generated model calls are set to closely match what you would see in nature.
One more question. Sorry, because I'm playing back my history of recording from these neurons. When you show that the exposed males have less decrement, is there also fewer spikes produced per chirp, so that it's a low number for both the first and third syllable?
So that's actually a really good question. It's part of the analysis component that I haven't been able to conduct as of yet. Right now, we just have percent work on that level.
Interesting, I think that's,
It would be interesting.
that's typical of the older females, right? It's lower response to both, so. Interesting.
Thank you very much.